I'm going to report on what we've done in the context of the OmicsDI Cloud project, which is one of the projects which Phil has just mentioned, which were organized by Work Package 1. And the aim of the project was to provide deployment of OmicsDI on EOS Cloud infrastructure to provide an open source code base for OmicsDI local satellite deployment so that we could clone the main instance with optional synchronization from the OmicsDI public instance. And then to demonstrate this by integrating additional resources into the Omics Discovery Index, and to also support further adaptation of the system through biohackathon or similar. Keep in mind, this was conceptualized before the lockdown. And finally, as somewhat separate deliverable, to provide infrastructure to allow third parties to use the impact metrics functionality which we've developed in OmicsDI. So this is only a very quick overview, and I'll explain more what each of these mean and what we've achieved in the following slides. So first of all, OmicsDI. What is OmicsDI? OmicsDI is short for Omics Discovery Index, and it's meant to be a kind of PubMed or Europe PMC for omics data. It integrates data from 23 different repositories currently across multiple omics types, proteomics, transcriptomics, metabolomics, etc., into one common discovery interface, and provides a view where with one search you can query across all of these resources and get the results across these resources, with the aim to also overcome the siloing, which is often caused by specialized resources, which can provide the best infrastructure for a specific data type. But it often requires that for multi-omic studies, you have to split your data up across multiple repositories upon deposition. And OmicsDI allows then to, again, re-establish the connection between these by through the shared PMC ID. So what we have done, or the aim, was to provide a cloud-based deployment of OmicsDI and a satellite installation for a reduced data set for systems biology. And this, we have worked on the Kubernetes-based deployment of OmicsDI in the EBI Embassy Cloud. Unfortunately, this is still in progress now. There was a technical upgrade in the Embassy Cloud, and which meant that we had to give up the original installation, and we have not yet brought back the new installation, the new deployment in the new Embassy Cloud Kubernetes environment up to production status. So that's still ongoing, but all the functionality is accessible through the, let's say, classical deployment on the EBI internal cloud. It's just what we can demo is not based on the Kubernetes infrastructure, but that is still to come. And that's what I will focus on now. So we have a use case from the systems biology community. In systems biology, there is a number of repositories which all do their own curation from the literature. They accept submissions of mathematical systems or mathematical representations of biological systems. And if you want to retrieve all relevant models across a number of databases, you have to launch multiple queries. And so the aim in model exchange, an informal consortium between a number of key repositories, is to provide a common infrastructure which allows to increase, in particular, the F part, the findability part of FAIR through a common search interface, but also in the longer run to coordinate curation activities so that the same data set is not curated, model is not curated multiple times. And in the context of this proposal, we have developed a satellite instance of OmicsDI, which provides a separate interface for systems biology, and biomodels was already part of OmicsDI before, but we have added 3 new repositories: the Physio Model Repository, Hermjakob, and Cell Collective. And these are now all available through the Model Exchange interface. And this supports now cross-repository search across all of these repositories. It allows to see, as you can see here, what are the latest data sets, again, jointly from all repositories, what are most accessed data sets in this interface, to really provide one search engine for at least the key repositories in the systems biology community. One of the use cases for this is redundancy detection. As I mentioned before, it is a fairly common occurrence in independently operating repositories that more than one repository might curate and provide the same model. That is not necessarily bad. It allows that you can compare different representations in the repositories. It allows some level of quality control by seeing if the representation in the different repositories is the same. But of course, it means a lot of additional curation work, and therefore ultimately perhaps not the best use of taxpayers' money. So in the first instance, we want to be able to at least detect if a model occurs in more than one repository. And we are doing that in Model Exchange via comparing or mapping all entries to the PubMed IDs they are related to, and then in any given entry provide links to other entries which reference the same PubMed ID as their source paper. And in this case, it's easy to discover that, wait, this model is not only available from, let's say, biomodels, but also from the Cell Collective. And then you can access also these alternative representations. And of course, as I mentioned before, this is only the first step. The longer term idea is that we will coordinate our work among the systems biology repositories to avoid the redundant curation. And so you can see here in this representation of this particular model, you have a link to the source of the model in the original repository biomodels, but you can also see alternative related representations, related entries which reference the same original publication in both another repository, Cell Collective, and also a second model which came from the same publication in biomodels. And then you have the classical, well, you can access the model in the original repository, but OmicsDI and by cloning Model Exchange also provide then direct access, also for API access to the underlying data files where we have these. And in other projects, we have demonstrated that this then supports integration into workflows, because you can, via API, discover relevant data sets and then directly access their data files. But that was done in a different context. Quite innovative feature which OmicsDI provides and which Model Exchange now provides through work done in this project is that we do a prototype implementation of impact metrics for data sets. And this is based on multiple dimensions, for example, views within OmicsDI or Model Exchange, downloads. This works currently only for EDI repositories, but of course could be extended in the future if we develop a protocol to transfer the metadata on downloads from different resources, citations of the actual dataset accession number, not only the related publication, but the dataset itself, and references among data objects. So for example, when an entry in a curated database, like let's say UniProt, references a proteomics experiment, or if one model, systems biology model, references another systems biology model as its original base model on which further development has been done, then we aggregate this kind of usage and impact metrics, normalize them, and then provide them in a kind of rosette, which on purpose looks very similar to altmetrics, to give at least, I would say, a nascent or prototype view of what the impact of a specific data set is. It's clear this is not a complete view. It's clear this is very exploratory, but the aim is to contribute to efforts that make data objects first-class objects of scientific output, and to ultimately value output in more than just the number of publications a scientist has done, and also value open data sets, curation efforts, etc. And so this rosette is accessible in Model Exchange and was accessible in OmicsDI before, but what we have added is an API which allows to include this rosette in third-party websites. And so now in two demonstrator projects, the PRIDE database and the biomodels database both provide these OmicsDI impact rosettes in their entries. The data is integrated via the API and allows to give this, let's say, notional idea of the impact of the data set in these third-party resources. And this can be used, of course, not only by third-party databases, but also in the long run could be used by authors, organizations, funders, etc., to include this in a view of the impact of data sets for various purposes. And I think we think that has a very important long-term impact on valuing data as valuable scientific output. You can see it here. It's quite small in this representation in the bottom right corner in the biomodels database. So these were the this was the work we have done in the context of the project. And now a bit on the specific questions which were asked. The impact of the project on research infrastructures, I would say on a technical level, what we have done had limited impact. We implemented standard Kubernetes deployment on the EDI Embassy Cloud. So this is software engineering, but I would say it was not particularly technically relevant. Our impact is much more on the community impact ultimately in the promotion of FAIR principles. We believe that the specialization of OmicsDI to model exchange may have pilot impact for other fields. The purpose of this clonable feature is that you could have other specializations of OmicsDI for other domains using the same platform, but allowing then a community-specific view of the data in OmicsDI. And it's very likely that model exchange itself will have a significant impact on the systems biology community because it for the first time enables a cross-repository metadata-based discovery, and a previous related effort, proteome exchange, contributed really to changing the data culture in proteomics from closed, just publishing lists of identified proteins, to open, where now it's really normal in proteomics to provide all your supporting raw data for further analysis. And the impact metrics for data are, I think, a logical follow-up to the FAIR data efforts. And once we have data that is accessible, clearly we also want in the long run to understand what the impact of each data set is. We probably won't be able to keep all data forever. And so methods that help to assess the relevance of data are, I think, something the community needs and will need to will invest into. And so that's a longer term, indirect, but I think fairly important impact. The impact of the project on EOSC, the multi-omics, multi-repository data, multi-omics, multi-repository metadata discovery and metrics may have a pilot function for EOSC overall. And the specialization of the multi-omics infrastructure to a specific community is probably a kind of simplified case study for the EOSC challenge, which needs to provide general infrastructures which are useful for specific domains without too much adaptation overhead. Our experience in working with EOSC-LIFE and the technical teams was, I would say, not too intensive, which was probably mostly a problem on our side. We have approached this as a technical project. The regular EOSC-LIFE meetings, which were offered, to which we were invited, were kind of just another meeting, and so, in all honesty, often skipped by me. And it was quite challenging to really look beyond the own immediate project into the larger EOSC-LIFE context. And that is probably part of having a limited specialized project in this large context. There's lots of large projects, and understanding and deeply engaging with all of them is quite challenging. And so I think we could have done more there, but I think that was more on our side than on the EOSC-LIFE side. We have focused on the technical level on the existing EBI infrastructure rather than broadening out and, for example, aiming to install the system on another platform. Of course, the aim is to enable that with the Kubernetes deployment. We have good experience of transferring between the EBI Embassy Cloud and, in the specific context of another project, the Google Cloud. And so that's the aim. But as I mentioned, that is not the aim. This is still under development. And in retrospect, it might have been better to, from the beginning, aim for a different platform. That would have been more challenge because we would have had probably more technical work and would have not reached that much in terms of the actual application side. So I think we struck a balance. We focused on getting community impact rather than the technical impact, but we hope that we can still catch up on the technical impact and get that finalized and into production status. The sustainability, I think, is quite good. We have thought about the sustainability beforehand. Although the Kubernetes deployment of OmicsDI is still ongoing, this will be the long-term deployment of choice in an environment where the trend goes towards hybrid cloud deployment within EMBL EBI, beyond that. And so we will get there eventually and have OmicsDI as one building on this future infrastructure and being maintained in this cloud environment rather than the more localized environment it's currently running in. Model exchange itself is an informal but very active collaboration of systems biology data resources. We have discussed the concept for a long time. The presented work is a prototype, but we definitely will sustain and further develop this together, and this will also support future grant applications. The dataset metrics I see as a prototype, as explorative work, but as something which hopefully will contribute to value in scientific output and in particular data output in the same context in which publications are valued and contribute to a more balanced view of scientific output overall. And so I think this is an important point to long-term development in data science, but clearly exploratory and not necessarily in this particular form, but as one stepping stone to valuing a broad range of scientific outputs. And with that, I would like to acknowledge Gauhari Das and Robert Petrashak, who have done most of the development work, Yasser Perez-Riverol, who contributed to the concepts initially, Tong Guayen from Biomodels, who implemented the Rosette view in Biomodels, Helen and Phil for advice from the Work Package 1 side, and contributors for the systems biology data, Wolfgang Müller, Thomas Helliker, and David Nickerson from different systems biology repositories. Thank you.
Thank you very much, Henning. It's a very interesting talk. It's very nice. I'd ask if people have additional questions, then please put them in the chat. I got a question that's in there. It's around your improved impact metrics approach. Do you see this as being sort of useful for non-OMIC data resources from the sort of life science RIs or even other research communities? Is it transferable to those domains, you think?
Absolutely. I see that as a main challenge we need to address across any scientific domain if we want to really continue and doing the next step after making data fair, of allowing to assess the impact of data, because that will ultimately be a major incentive of providing the data publicly. And we probably can't have all data forever available, but if we don't know what is used and what is not used, then we can't make informed decisions. So we all know that bibliometrics like the H index are not exactly great. They provide a very one-dimensional view of scientific output, but they are still one important part of assessing what the value of scientific output is, and why we definitely want to have more than one number, to at least have a way to collect evidence on what impact a data set has, I think is something which needs to be done across domains. And I think the methods which we are using here are transferable, almost all of them in concept to other domains. So cross-referencing between data sets, how one data set builds on another one, how a reanalysis uses one data set. I think these are all perfectly